crowd here, and thank you for coming. I would like to start by mentioning why we have this event and how. So we have good friends, Moveron and Michael Heller, who are a couple from England, London. In 2002, they decided to establish at LSEC the Heller Lecture Series in Computational Neuroscience. 2002. This was, exactly, this was at the time we were ICNC, the Interdisciplinary Center for Neural Computation, and we continue to be a center for computational neuroscience. Only the terms has changed a little from neural computation to computational neuroscience, but it does mean something. We are not neural computation, we are computational neuroscience. There is a difference, and you think what the difference is, but the computation stays at the core of our work. The primary aim of this unique lecture series was to acquaint the interested public with most exciting research advances in brain sciences, and I'm happy to see that the public this time is public of young students who came here in tens and many. So for every year, 4 renowned scientists visit the Hebrew University, deliver talks, interact with the students, and they go to dinner with them, etc. And they meet the members of the center and so on. So today we are proud and happy to host Matteo Carandini, a world-renowned scientist who is the Heller lecturer of today. Matteo, as you could guess from the name, and when you hear him, you will still hear the reminiscence of this, was born in Italy. He received a PhD in the United States and then continued in Zurich and back to the United States until he moved to UCL, where he's currently a professor of neuroscience and working in computational neuroscience, I believe with collaboration of Gatsby, etc. So together with David Higer and Anthony Mulshon, Matteo and other colleagues developed what is called the normalization model, which significantly advanced the quantitative modeling of visual receptive fields in retina and cortex. Carandini addresses fundamental questions in neuroscience, combining theory, computation, and experiment with focus on visual processing, but questions that are much more general. For example, there is a paper called, for those who are interested here, 5 key factors determining pairwise correlations in visual cortex. This was in 2015. But reading the title of today's talk, I guess that Matteo will include findings from a study of his group published in a Nature paper of 2015, where the first author is an Israeli student whom I personally love very much, Michael Okun, who was a student of Ilan Ampel and worked a little bit in my lab. So I think he will tell us about his new ideas today, Matteo. You will tell us about new ideas about social hierarchy in the brain and various personalities of neurons in this kingdom called the brain. Thank you very much for coming, soloists and choirs and choristers in the orchestra. Thank you very much.
Thank you for having me. How is the volume? Can you hear me okay? So I know time is precious, but I need to tell you 2 anecdotes. The first is that it's really great to have spin glass theory equations behind me, because my first contact with neuroscience if we can call this neuroscience, by the way, was by being a student of Giorgio Parisi, who's a theoretical physicist in Italy. And I was a student of Giorgio Parisi for a very short time because he had a filter method, which was he would give people a book called Spin Glass Theory and Beyond, which at least contained a very big article by Amit Gutfreund and Sampolinsky, and said, read this and come back. I tried to read it, I never came back. And the other anecdote is that I was introduced to some of the key people in this talk. Again, very early on, Misha Toduks had, as many people, left Russia and came to, eventually Israel by way of Italy, which was a common thing in the late 80s. And so I was in charge of translating from Italian into English to Misha Toduks. And I don't know if you know Misha, he's always been very quiet, and he was very quiet, so I never knew if he understood what I said. Probably due to my English. Anyway, thank you so much for having me. This is supposed to be a lecture that is understandable also by people who are not neuroscientists. So I would be super happy if you raise your hands every time I say something that is not clear. If instead you're raising your hand because you disagree with me, please don't raise your hand, because you'll have another chance. I'm giving another lecture in 2 days on slightly different topics, but that's a neuroscience lecture, and please do disagree with me there. Okay, but not here. Can we dim the lights? Is it easy from here? Okay, so my title is Soloists and Choristers in a Cortical Population, and given that there's lots of young people, let me just remind you that this is an orchestra, and that there are some people who are soloists. They sing their own thing, hopefully in some kind of harmony with the rest. Some of the violins also might be soloists. This is definitely a choir. They all sing together, maybe in groups. And then different instruments also play in groups. So we'll use these words soloists and choristers, but I feel that sometimes it's useful to define these things because a lot of people have never seen an orchestra. Anyway, I know in Israel this would not happen, but anyway. Okay, so broadly speaking, there's many ways to make these cartoons about what we try to do in neuroscience, but one way to think about it is that we're trying to understand how a brain generates behavior. I come from a tradition of sensory neuroscience, so I always think of stimuli coming in this case, visual stimulus coming into a mouse eye. And so I think of it as going this way. But you can also think of information going the other way. And we know that the activity of neurons and populations of neurons, even in sensory parts of the brain, are heavily influenced by what the mouse is doing and what the behavior is, etc. So what I'd like to do is give you an overview of what I think are exciting new methods to study various aspects of this cascade, if you want. And then I will focus on a scientific question that is here between neurons and populations. And you can think of it as this being the orchestra and maybe these guys being soloists or choristers. And so part of the talk will probably be too broad for some of you, and part of the talk will actually probably be too narrow for some of you, so I apologize to both. And so let me start broad and tell you about methods that we have in neuroscience to study the activity of neurons and populations. The most classic one, which has given rise to a good 60 years of amazing neuroscience, has been the microelectrode. This was a major technical achievement in the 1950s, a tiny piece of metal that is very thin and has only a tiny bit that is exposed and allows you to pick up electrical signals from only one neuron if you put it near a neuron. And this has told us an incredible amount about how the brain works. In the last 10 years or 15 years, instead, we're using more and more of these multi-site electrodes that have multiple sites where you can record electrical activity. You don't have the pleasure of advancing them until you get to a nice big fat neuron. By the way, from the neuron you would record these electrical events called spikes, and usually with microelectrodes you would advance the electrode until you get a big spike. Here instead the sites are where they happen to be, and you happen to record from the neurons that happen to be around. You don't get to pick and choose. This, by the way, leads to much more unbiased estimates of what the neurons do, because you're not choosing, today I would like a neuron that does this. And this method keeps on progressing, and so there's new technology coming out, and this is going to be available commercially in 2 years, in principle, out of a big collaboration that involves Allen Institute, Janelia Farm. These are big neuroscience institutions in the US, the Wellcome Trust in England, and our lab is just part of that collaboration. There are new electrodes that are coming out that have 1,000 sites. So they end with a little tip, which is magnified here, and then there's 500 sites on this side, 500 on this side, and here I'm only printing a few. The picture would keep on going to the other floors if you have them. And out of when you have these long electrodes, a neuron might be here, for example, and might give you signals on these 4 or 5 sites. For example, take the neuron that is in red here. You see that it's giving a certain electrical waveform. This is a spike that appears, say, in this site, this site, this site, and this site, 1, 2, 3, 4, and next door to these 4 sites. But another neuron will give rise to another combination of activity, and so it's an interesting mathematical problem how to unmix these signals, right? But once you solve that problem, and solving is a big word, but you can get close to solving, you can record the activity of lots and lots of neurons. Here you see the electrical signature of 1, 2, 3, 4 neurons, and then we can try to estimate very roughly where they are. In this dimension it's easy, in this dimension it's hard, and in this dimension it's really hard. And so you can imagine that this little chunk of a ray will be recording from all these neurons colored this way. And if you would like to see the electrical signatures of all those neurons, here they are. There are just too many to put in the graph. Okay? So this is one electrode. And this changes the life of the electrophysiologist because this electrode does everything on board. Out of it literally comes a USB connection that can go into a laptop. The rest of the lab becomes unnecessary, essentially. So this is the future of electrophysiology. And so using this kind of technique, but even the simpler multi-site arrays that already exist, this keeps on going, by the way, it's not looping yet. So it's just one electrode, more and more neurons. You could take a lifetime to get this many, one at a time. Okay, using this kind of techniques, I would like to tell you about one story that was developed by Michael Okun, who was first a computer scientist and then a neuroscientist, thanks to Israel. And he's been a postdoc in our lab, and now he just got a job in Leicester, near London. And he approached a question that is a question that started entirely here in Israel. And it was a question posed by these scientists, Amos, Arieli, Misha Tzadok, Miriam Greenwald, and Talkenet was a student there, I believe. And they asked this question, are single cortical neurons soloists, or are they obedient members of a huge orchestra? This was valued as question number 9 in this book. I think it's number 1, but hey. And in typical Israeli fashion, they also had an answer. And so their answer was that they are members of a huge orchestra. And we can talk about why they gave that answer. But I want to give my answer. Or rather, Mush's answer. So to address this question, Mush recorded from a lot of neurons at the same time in the visual cortex of a mouse that is in this particular moment, for this slide, this mouse is looking at nothing, at a gray screen. Okay? And you can follow the activity of the neurons. These dots tell you when the neurons fire. This is time, and this is neuron number. And I don't know how many neurons there are here, maybe 40. And then this is simply the sum activity of all these neurons over time. And we'll call this the population rate. We have arranged these neurons in order of how correlated they are with this trace, with the overall population. And this one is one of the least correlated ones. We're going to call it a soloist. These guys here are more correlated with this. We're going to call them choristers. And don't get too attached to some notions you might develop. For example, you might say, oh, the soloist fires very rarely. The key point is not how much they fire, but the fact that they fire at times that are unrelated to when the rest go fire. Okay? And to measure this quantitatively, you can measure the correlation between the firing of one of these neurons and the population rate. And as you can see, the red one, which I had called a chorister, has a very high correlation with the population. By the way, the traces are way longer than this. You're just seeing a little bit, okay? And the soloist has zero correlation, if anything, something negative. It almost does the opposite as the population. Okay. And the intermediate ones are intermediate. By the way, these words, like soloist and chorister, are words that are useful just the way you would describe somebody as being tall or being short. It doesn't mean that there are two discrete groups of people, half of which are tall and half of which are short, okay? So we don't think that there is a continuum. It doesn't mean, these are just words that are helpful to talk about that continuum. So the first thing I'd like to show you is that just like real people who play in orchestras, soloists and choristers don't change jobs. If you record from a mouse that is looking at nothing, and you follow those 4 neurons, and you record from a mouse that is looking at a natural video, by the way, this should be black, I apologize. These are the same 4 neurons, or rather, this should be purple, because it's these 4, 1, 2, 3, 4. You see that they have very similar correlations with the population. So this guy was a soloist when the mouse was looking at nothing, and he remains a soloist when the mouse looks at a video, and so on. And by the way, these neurons have very nice properties that you would expect from visual neurons. For example, selectivity for orientation and other things that would be normal in the visual cortex. Yes?
So you consider a soloist also a neuron that's anti-correlated?
Yes, although that's rare. You will see a plot of correlation at some point, and where you will see blue, that will be negative.
So you don't distinguish correlation?
No. Okay, so first observation is that the soloists and choristers don't switch. They might switch over longer timescales, but over a few days, over actually a few hours, they don't switch. Another observation is that the choristers tend to be more responsive to visual stimuli. So here is the red one, which was one of the more choristery ones. When a visual stimulus comes on, and it doesn't really matter what visual stimulus, it's a natural movie, this neuron makes more spikes. And you can see the firing rate of the neuron goes up, above the average. And the soloist instead still makes spikes at particular times in the movie when there are visual features that the neuron likes. But overall, the overall firing rate doesn't change. And this is a trend that is consistent. So neurons that have high population coupling, so more choristers, tend to increase their mean firing rate when you show a visual stimulus. And neurons that are more soloists tend to either not change it or even decrease, depends if you believe in this or not. And so there's a trend there. The neurons that are more choristers tend to increase their overall firing rate with visual stimuli. Now, this might make you think maybe the choristers are somehow more connected to the rest of the network. And maybe that's why they correlate with the rest of the network more. And so what I'd like to do is first give you a sense of causality there, because correlation is non-causal, right? You don't know if correlation is due to causality. But what we're going to do now is to make some neurons fire on purpose when we want them to fire, and we will ask what happens to the choristers and what happens to the soloists. And this experiment is done using a technique called optogenetics, in which you take a mouse that expresses a certain protein that is not supposed to be in brains at all. It's a protein that responds to light. This is a piece of the cortex of the mouse, and there are neurons in layer 5, which is one of the layers of the cortex, that express this protein, and they happen to fluoresce. That's why you can see them. And when you project light on them, they fire. And so now the question is, what happens to the neurons to the other neurons when you make these neurons fire? And what happens depends on whether neurons were choristers or soloists. So again, this is a lot of neurons. These are 4 instances in which we're shining light to drive these neurons. The classification of these neurons was done when the animal was not being stimulated in any way. So this is not a circular argument. So the neurons have been arranged. Classification is the wrong word, but the ordering of these neurons has been made previously, before this experiment. And now you can see that on average, when you make these neurons fire by shining blue light, which is in these periods, the neurons that are more choristers tend to increase their firing rates, and the neurons that are more soloists couldn't care less. And that's illustrated here. The choristers tend to increase their firing rate. The soloists don't care. Okay, so but what have we demonstrated? We demonstrated here that the choristers are somehow more functionally connected and more causally connected, but that still doesn't tell you much about synapses. I don't know how many of you here are of a biological background, but people who study, you know, the nuts and bolts of the brain would like to know about actual synapses and actual connections. And so to study those, we teamed up with a team who used to be at UCL and then moved to Switzerland. It's the team of Tom Merzic-Flogel. But again, this is all Michael Okun collecting data from a lot of sources and coming up with a coherent picture. So it's, this is data that already existed. This group in Switzerland had performed these really interesting experiments in which they first imaged the activity of a lot of neurons in visual cortex, which is up here, using a technique called two-photon imaging, which I'll describe in a minute. And then what these people did is they sliced up the brain and put electrodes in as many neurons at a time as they could, and then established who was connected to who. And they had this data on their hard drives. So what Mush did is he went back to these neurons in their data and asked who was a soloist and who was a chorister of these neurons. And he found that some of them had high population coupling, the ones shown in red. These are more choristers, they're more correlated with the overall activity, and some were more soloists. These are shown in blue. In this case, it's all positive, the correlation. But, let's call these soloists, okay? And you can see already that there's no particular structure to whether you're a soloist or a chorister. But then, when he compared this measurement of population coupling, which is shown here, to the measurements of connectivity that these scientists had already made in a slice of brain, he discovered this relationship, which is that the more a neuron is a chorister, the more it is likely to receive synapses from the others. The fact that he found this relationship is very remarkable because this sampling, this is a major undersampling of the actual connections in that circuit, right? Because these scientists had limited time to poke a bunch of neurons, and so they severely underestimated the number of connections that are actually in there. And so the fact that even with this severe underestimation, we could find this relationship, it means that there is actually probably a very strong relationship down there in the brain that we actually don't have the data for. Okay, so to recap up to here, we have these neurons that are more connected physically to the others. They receive more synapses, and they correlate more with the group. You could say, yeah, it all makes sense. It's kind of intuitive. It's cute that you've gone from correlations to synapses, but is it useful? You know? And so for some of you, useful means, does it explain a large body of data? So for that, I need to find a way for you to ask questions if you're not understanding something, because otherwise I'll keep on giving my regular neuroscience talk. I doubt that you didn't understand something, Chaim. But anyway.
For bit of connection, what about the strength of connection?
We don't know. We did not look at that. By the way, these are all excitatory neurons. We did not look at the strength of connections. It is in some of their data sets, but not in some others. So, yeah. The ones that were recorded electrically, not necessarily. And we found really faint relationships with whether a neuron is inhibitory or excitatory and whether it's a soloist or a chorister. Okay, so if you're recording from a lot of neurons, and here comes the answer to your question, and you want to characterize their overall activity, one thing you may want to do is characterize their pairwise correlations. I bet that somewhere in there there's pairwise correlations. And so it's just one of the statistics you may... so it's the second statistic you'll come up with. The first statistic is their mean activity, and I showed you that it's called the population rate. Now let's move to the next statistic, which is the correlation between two neurons. And so this is data. And it tells you that, so red means high correlation, blue means actually slightly negative, white means no correlation. And this matrix is symmetrical, and there would be all ones here, so no point plotting those, okay? And so you can see that there's some kind of rule, which is that the choristers tend to be positively correlated with choristers. The soloists happen to more often have some blue, some negative correlation with the rest. And I wouldn't read too much into having more blue here or more blue there, because it depends on the data set, okay? But the general idea that you go from red to blue is fairly common. Yes? No, that's the point. I would not argue that it's bimodal. It's unimodal. And that's why I made that example about tall people and short people. I really doubt that the distribution in the world is bimodal. It ends up looking like a Gaussian, but that's because of the way we pool the data, which we always normalize them, and basically we're descoring all of them, and so if you do it enough times, you end up with a Gaussian. Okay, so now the question is, can you predict this fine structure? I'm not talking now about the obvious structure from red to blue, but the fact that there's a red dot here and a blue dot there. How much of that is due to exquisite wiring properties of these neurons, and how much, specifically of this neuron being specifically very precisely wired to that neuron, and how much of it instead can we explain based on this low-order explanation? So let's start first of all, with the low-order explanation being soloists and choristers. Let's first start with an explanation that is not going to work, which is an explanation that simply takes into consideration two things. One is, how much does a neuron like to fire? And the other is, how likely it is to have a certain firing rate in the overall population. So this is going to be our null hypothesis. First of all, let me show you, it does a bad job. And here I'm counting on your visual system's ability to detect symmetry, and in this case, to not detect symmetry, because this does not look like that. Okay. And the model that we're using here is a model that Mush actually had proposed the year before for something else. It's a model that says, for each neuron, I know when it doesn't spike, and I know when it does spike. So this is the word, basically, pronounced by that neuron. This is the word by another neuron, the word by another neuron, the word by another neuron. And if I only know two things, how likely a neuron is to fire, which is its mean firing rate, and how likely a certain population rate is, which is the distribution of population rates, can I predict the pairwise correlations? And the answer is no. But look at what happens when you add a third piece of information, which is how much each of these is a soloist or a chorister. When you add that piece of information, you do a pretty good job. And I'll show you. Will I show you? I'm not sure that I'll show you. It's in the paper. I would have shown you in the paper a graph of when we do a good job and when we do a bad job. But what you can do now is use your visual system and notice these symmetries. Okay? In some cases, it's a really good job. In some cases, it's a bad job. And it turns out that you do a particularly good job when the population rate changes a lot, which is intuitive. If the population rate is super flat, you do a bad job. And so you can predict a lot of the covariation of two neurons from this really simple concept of population coupling. Okay.
So you say when and when, you mean
animal to animal. Animal to animal. And again, for those of you who like to have a take-home message in terms of numbers, this is order n square numbers, n neurons by n neurons. And this is order 3n numbers. And the nice thing about these order 3n numbers is that they have very clear biological meaning. How much does a neuron like to fire? How much does the population like to fire a certain amount? How much of a soloist or a chorister is each neuron? They have measurable and intuitive meanings. Anyway, this is the work by Michael Okun, and I want to summarize it as follows. Population coupling, which is this idea of whether neurons are soloists or choristers, is an invariant property of neurons within the couple of hours of our experiments. Okay, we don't know if a neuron, maybe after this talk, some of your choristers have turned into soloists. I don't know. Okay, but it predicts a bunch of things. It predicts visual responsiveness. It also predicts top-down involvement, but I didn't show it. It predicts functional and causal connectivity. It predicts anatomical connectivity, and it really helps you get a hold of order n square pairwise correlations. Yes. The animals were awake. Okay, so what? Yeah.
So do you have a generative model how to?
Yeah. So Mush has, from the computer science literature, he can produce matrices that obey these constraints, and they come from the computer science literature. You would have. I can tell you what they are, but I would have to ask him. Yes. I'm going right there. Because I just gave you a really clean story, I'm going to make it a little more complicated. That's exactly where I'm going. I'm going to show you that there are cliques of neurons. And I'm getting there. One more question? No. Okay, so now I'm going to go back and being a bit of a generalist and tell you about other methods that we use in neuroscience. I do realize that many of you here are neuroscientists, so you don't need this, but it is fun nonetheless. We're now going to move to two-photon imaging, and this is an example of imaging of a single neuron in the visual cortex of a mouse. The neuron expresses a calcium-sensitive fluorescent protein, which is called GCAMP, in this case it's GCAMP6, and this protein fluoresces the more the neuron has calcium. And the more the neuron fires, the more it has calcium. This particular neuron was injected in vivo in the middle of the head of the cortex of a mouse. And you can see now all of its spines and dendrites, and this is the little section, and you can see that if you look at a particular spine, you'll see the calcium in that spine, but you can also use it to measure the activity of the soma. But this is just to demonstrate the technique. Now I'm going to zoom out and go to a bunch of neurons. And, if you follow a bunch of neurons, they fire every once in a while, I don't know. This video is too compressed. It looks really ugly from here. I hope it looks good from far away. So anyway, you can see these neurons firing. Now, Marius Pachitariu is a brilliant postdoc in our lab. He developed, I think, one of the winning methods for selecting these neurons. Now, we zoomed out even more now, okay? We were looking at a little region. And identifying neurons on the basis of correlations with the nearby pixels, okay? And so he has this method that, out of an image like this, can give you about 1,000 neurons, which you can then follow one by one.
So
yeah.
What is the special scale now?
It's about 1 millimeter by 1 millimeter. This is an important question, because what he does next is he's going to go up or down, which means up or down in the brain, by a total of 300 microns. And he's going to take 11 planes like this. And I will show you some of them. And by doing this, he can record from 14,000 neurons. He does have to give up a little bit on temporal resolution, because he needs to do this at 5 hertz, which means he uses a sensor, a calcium sensor, that has slow decay, called GCAMP6 slow, so that if a neuron spikes, even if your two-photon microscope is not on it at that time, you can still see the calcium of it. Okay.
You don't mean a single spike. You mean if a neuron there is a calcium burst. There is
Yeah, actually, there is, the people who. Okay, exactly. So now, there's a couple of interesting questions here. First of all, if you can record from 14,000 neurons, are you going to discover anything that you would not have discovered if you recorded from 500?
Yes.
Well, we don't know that, right? Nobody has recorded from 14,000 neurons before, as far as I know. Number one. And number two, what can you learn about the brain once you've done that? So, let me show you a few of the results. These are super recent. I take the liberty of changing my mind on the next 5 slides. Efforts taking place in my lab and in the field to study the brain at this larger scale, because the challenge, I think, one of the challenges is to understand how large populations of neurons not only work within themselves, but also how they work together to achieve behavior. And so I just want to show you some cool techniques for probing the brain at this larger scale, and some techniques for probing behavior. And you can imagine that when you put them all together, which is where the field is going, you have a nice synergy. So let's go to the scale. And just to refresh you, this is the cerebral cortex. This is the cerebellum. We don't look at that ever. And this is the olfactory bulb. It doesn't even deserve a color. There you go. And so this, the cerebral cortex, is what really matters. This is the basic plan for the cerebral cortex is the same in all mammals. Only mammals have a cerebral cortex, unless you want to change some concepts of bird physiology, which might be okay. Visual cortex is back here, shown in blue. Auditory cortex is on the sides, shown in red. Somatosensory cortex is here, and is shown in green. And in the rodent, the barrel fields are particularly strong because they're associated with the whiskers. And then there's motor cortex, and then pretty much, this is pretty much all they have, frontal cortex, which for us is, from here on. And so, but they have all the major pieces. Now, what we have been doing is image an entire hemisphere, or almost an entire hemisphere. And this is, you can see that we stop around here, using, in this case, voltage-sensitive fluorescent proteins. And I just want to be quick about the technique, but the technique is Whitefield imaging, which means we do not have resolution at the level of neurons. Now we have resolution in the order of 50, 100 microns. And here's a simple experiment in which we have a mouse, we stimulate the whiskers, we see activity here, which makes sense. We give tones, we see activity here, which makes sense. And we give visual stimuli, we see activity here, which makes sense. Okay? So I'd like to give you a sense for what this activity looks like. So let me click on this video. Is it okay if I talk another 4 minutes? Yeah? Okay. So this is what the activity looks like. Slowed down 5 times. This is the visual response to a bar moving in front of the visual field, and you can really follow the bar, okay? And the bar goes at really fast, by the way, and this is why it looks like that when you slow it down. And if you what did I want to say? If you study the phase of the position of the bar at every moment in time, you obtain this map here, which is a map of the different visual areas and their retinotopy. I don't know if you can see down here. There's a colorful map down there. Not crucial, okay? It just explains to you why we have these contours. Now, what I'd like to do is something that Amos Ariely and Miriam Greenwald and all these, incredible scientists have done in the 90s in cats. Let's just do it again in mouse now, but looking at a broader region. Let's look at ongoing activity without a visual stimulus. And, sorry, I need to stop this because it gets confusing. How do I stop it? Well, sorry, I'll have to confuse you. This is the ongoing activity, and it has the same scale. It's the same, actually, you don't even know what the scale is. Sorry, I forgot the numbers. But anyway, it's the same color bar, okay? And
the scale you mean spatial scale?
Sorry, no, intensity.
Yes.
You see? So ongoing activity, when a mouse is looking at nothing, and maybe it's, moving a whisker, scratching its bum, whatever, it's doing stuff, okay? Awake, yeah. It has the same kind of feel and amplitude and spatial scale and temporal scale as driven activity. And what I'd like to, and this was known. What is less known, and by the way, understanding this is a challenge, okay? But it's a really important challenge. But what is less known is what happens when you look at this activity in the other hemisphere. And I think this is going to be amusing. So now, this is in the last couple of months, we started doing bilateral imaging. So we have this sort of triangular exposure on the brain, and this is visual cortex on the 2 sides. Our lab, in principle, is a visual lab. That's why we always have visual cortex. Now, this is calcium imaging, the same sensor that we use for 2-photon imaging. If we can make the room even darker, is there a chance?
Yeah.
So,
all the time. What's the time scale?
Now it's real time, I think, because this is time in seconds. I think it's real time. What do you think?
Yes, it is.
Looks real time. Okay, so this is, there have been papers saying this. We didn't discover this. Oh, let's start it again. It tells you that the ongoing activity, this animal is not looking at a visual stimulus, not doing a task, blue means less active than usual, than the mean, and red means more active than the mean. This kind of measurement is hard to assess zero activity, okay? And so you can see that ongoing activity has this beautifully bilateral structure. And there's been a beautiful study by Tim Murphy showing that if you take a mouse that is congenitally lacking boson that links the two hemispheres, the ongoing activity is no longer bilateral, which I think is a beautiful finding. It means that this is not driven by some subcortical structure. Anyway, so what we're trying to do now is understand the logic of this and understand how it interacts with activity here. And you can imagine the trick to do it. You make one piece of cortex fire by showing a visual stimulus to, for example, here, and then you ask how the activity here is due to the driven activity and to the ongoing activity. And that's what we're trying to do now. Okay, I really need to show you a couple more things really quickly. There's no more science, which is techniques that are being used in the lab, in my lab, and many other labs, to assess behavior. We started from a technique that looks like this. A mouse can walk around. If there's a grating on the right, it should go on the right. If there's a grating on the left, it should go on the left. And sometimes they make mistakes. The grating is on the left. That's a mistake. And now the punishment is trying to start the car without succeeding. And now grating's on the right, mouse goes on the right. Okay, so, and when you have this kind of task, you can measure the ability of the mouse to go right, depending on how much contrast there is on the right or how much contrast there is on the left, which is called the psychometric function. And you can imagine that if you did this during recordings of what's happening in the brain, you could understand the relationship between perception and brain activity. But you could also imagine that this is a terrible task to do that, because you don't know where the eyes are pointing, you don't know exactly where the head is pointing, you don't have, the brain is moving, which makes it really hard to record. So we've changed this task, and now we have a version in which the mouse essentially turns this little wheel. Okay, and now it's under a 2-photon microscope. Okay, and so the mouse licks when it gets a reward and turns this wheel to the left or to the right. But again, you can say, well, this task really is a very passive task. When does it happen in life that you have two stimuli, you need to choose one? Maybe a more natural thing for a mouse is to simply navigate an environment. And again, you can do that thanks to virtual reality with techniques that allow you to, change the visual world while a mouse traverses it, essentially. And there's various versions of this. And I just want to show you one more, and that's the end of the talk. And it's one that Michael Krumin, who is Lithuanian and then become Israeli, or maybe he was Israeli from the beginning, I don't know. He has a Lithuanian passport, that's what I know. Is studying now, and it's the interaction of vision, decision, and navigation. So here's a mouse that has a corridor. There's a grating on this side. It's hard to see. And the mouse goes there. Good. Then start again. Grating is on the right. Mouse needs to go there. Good. This is carefully selected, so I can say good. And at my next talk in 2 days, I will talk about how the brain responds during this task. I want to conclude by saying that clearly we're trying to put together these various levels. This is what the community is doing. We're not ahead of the community. I think we're part of the wave. The tools keep on improving. I don't know if they have reached sort of a steady state now, and we get these tools for a few years. Like the microelectrode arrived in the 50s or 60s, and basically that was the tool for a few decades. I don't know if this set of tools might actually stabilize for a little bit, and now we get to ask questions about how the brain works, okay? And so I had names down here for the people who did the work, so I don't need to thank them. I need to thank Simon's Foundations and Wellcome Trust because they fund us, and I also need to thank you for your attention. Thanks so much.
Before you ask questions, I would like to end this lecture this certificate, I don't know how to read it. The Heller Lecture Series in Computational Neuroscience award with this certificate recognition and appreciation to you, Matteo Carandini.
Thank you.
Very much.
Wow. Okay. Wow. Thank you.
And now the floor is open for questions. Yes.
So is it surprising or not that the peaks are roughly, they seem roughly the same size?
You know, don't read too much into what I said, that there are around 100 neurons. I just wanted to give you an order of magnitude. Is that what you're saying, number of neurons per click? Because we're still working on this.
Not the absolute number, just the fact that it's kind of uniform. That the peaks are roughly the same size.
I didn't say that. I didn't say that.
Mati.
But you found at least two, I think, things that were special for the choristers, that their responses were stronger and their firing rate, right?
Well, they elevated the firing rate. Yeah.
But on the other hand, you said that the soloist could be clicked as part of other. How does that?
We're still working on that. We don't know. I think the cliques. Okay, I had a slide. I took it out. The cliques tend to be overall more of a soloist than of a chorister. If you take an overall clique, it acts as a soloist. It tends to do its own thing. Like a clique of friends, probably deviates from what the rest of the group does.
So they're like orchestras of clicks, and
Yeah, that would be the intuition.
and maybe related to that, is there any, or did you look at the relation between these and the behavior? So
No. We just started with exactly what we want to do. We're putting that in our next grant. It's exactly what we want to do. Yeah. I don't know what.
Okay. Yeah.
Yeah, so this is what we're trying to understand. Intuitively, we're finding the neurons that are in cliques are more soloists than average. That's a fact because I checked it. And, so the choristers, essentially, you can think of them as belonging to one big clique, which is all the neurons. I'm saying this thing as if it was a demarcation. Okay, you're either in the clique or not. Clearly, this is smooth, right? But the idea is you can think of the choristers as belonging to one big clique, which is the overall population. As you record from more and more neurons, and you record from the whole brain, this concept of soloist and chorister would stop making sense, right? Because right?
It's different groups.
Yeah, I mean, it will stop making sense. You really don't want a neuron that correlates with the overall activity of the entire brain. So I think that's the correct way to think about it. That's how I would think about it.
Wouldn't this say that there is a relation between the location in the brain and what sleep you are in? Because you looked at one field.
Yes. So the
sources of visual cortex, the piece of visual cortex that you recorded, are actually part of a clique with.
That's what I think. Okay, so when we go in with 4 chunks of an electrode, we record from only, say, 150 neurons, roughly speaking, okay? Maybe fewer than that. In that case, what you find is a bunch of neurons that do similar things to the overall, and a bunch of neurons who seem to do their own thing. If instead of recording from around 100 neurons, you record from 14,000, you discover that the neurons that tend to do their own thing actually could, they don't have to, correlate very much with other neurons which you would have had extremely few chances of recording if you only had 100.
So if you have a distribution of click sizes on a like power line, I guess.
Okay.
So you would have essentially what you call the chorus, the largest.
The largest click. Yeah, I think it's this way, but we still.
From the many other things in the
But there might not even exist soloists. It might have been our. It must have been our mistake. Yeah, but they might not even exist in reality. Okay. The choristers tend to have stronger pairwise correlations with other choristers. That was in that triangular matrix, which I think is a necessity, by the way.
The other question I had was, some people have found it fine about different kinds
Yeah.
And their correlation with up and down states, whether they're
Yeah.
somatostatin or TV.
Yeah. So we were disappointed in not finding any relationship between what these measurements were and being interneuron. But we only were able to label interneurons from the spike shape electrically, and that's only PV cells. The jury is still out about somatostatin, VIP cells, and this whole story about alternating. Those were all excitatory neurons, I should tell you. The two-photon imaging, there were only excitatory neurons, only neurons that are in layer 2-3 and express CAM kinase 2, so excitatory. But that story needs to be worked out.
Nora?
Maybe taking the analogy a little bit too far, but do you have any idea of the conductor of your might be?
Well, I would have said no, but I can tell you who the manager is, because when the pupil changes size, it seems that an entire new orchestra walks in. So there's a manager that says, okay, Tuesday you're working, Wednesday you're working. So that's what if we want to push it that way. I think alertness totally changes who's up and who's down.
Alertness is caused by the solist or the orchestra?
I don't know.
And when you talked about the correlation between the neurons from which you derived your notion of cause and fluid,
Yeah.
you only looked at the same time step, right?
Yes.
And did you check whether there is a correlation between cause and fluid in the next time step?
Well, the correlations seem to have a fairly typical width in time, and they just seem to peak near zero. And we didn't do correlations with matrices with different time steps. No, we didn't see that.
You could have found maybe not the correlation, but you could have found cause fluid all the other time.
We only looked at time zero, delta time zero, so we didn't look.
More students? Okay. 1, 2.
How would you, with your addition to click and chain,
First of all, we were able to do this. It's funny because, we had to record for a long time because we were only going at 5 hertz. And so we needed to record for a long time, but we're very thankful that we recorded for a long time. And so we have a couple of hours of recording. And so I don't know how the duration of the recording would influence your ability to discover the cliques. Certainly, the longer the recording, the more 2 cliques who seem to be the same could deviate. And so we don't have answers to many of the questions you have. And I'm sorry, it's the price I pay for showing you something really recent. So I don't think it's wrong. I stand behind it, but I think some details, by the time we write up the paper, might have changed.
What would you expect to find with stimuli presented to the mouse? Does click code a stimulus or stimulus coded by?
Yeah, so this is the first thing we want to look at. As far as we can tell, the cliques have nothing to do with orientation selectivity. So, because you could imagine, a bunch of neurons that like this orientation, and they just, at some point in the darkness, they hallucinate this orientation, and they all fire together, which would be a strange thing, because they would be hallucinating it for like 50 seconds. We're talking about really long time scales here. And so the only thing I can tell you is there doesn't seem to be a relationship with orientation selectivity, but there might be a relationship with other properties that we still need to study.
All right. I think I will. Marcel, you still? Okay. I think because we have other events this evening, we will have to stop here. On Thursday, Matteo will give us another lecture, and we all thank you for this fascinating talk.
Thank you.
Thank you very much.